Isang magandang umaga sa ating lahat. The topics for this morning is all about the enzymes. So the second topic for biochemistry too. Okay, the enzyme is a biological catalyst, as I have made mentioned during the topics about protein. Okay, enzymes are basically protein. Okay, so this morning, these are the learning goals. Describe the role of enzyme and why they are essentials to living organism. List the factors that may affect the rate of an enzyme catalyzed reaction. Describe the importance of the structure of an enzyme to its functioning. So let us first discuss what is an enzyme is. Enzymes are molecules that act as catalysts to speed up biological reactions. So these are catalysts or to speed up biological chemical reactions uh, to uh, to speed up chemical reaction in terms of metabolism, in terms of absorbing nutrients to the different system of the body. The compounds on which an enzyme acts is a substrate. So kung saan nag-act ang enzyme is always be reacting to substrate. Enzyme can break a single structure into smaller components or join or two or more substrate molecules together. So, ang enzymes in our system ay nagbe-breakdown ng ating biomolecular components. For instance, yung carbohydrates can be breakdown into monosaccharides, yung ating proteins can be breakdown into amino acids, yung lipids can be broken down into its simple form. So, ang enzyme is responsible in any metabolism. Most enzymes are protein, as I have made mention. Okay, so this is an example of you can see here an pineapple. Okay, so lahat ng fruits contains enzyme. In this example, we have the pineapple or the Anaнаcomosus contains the enzyme papain, which is used in meat tenderization process and also medically as anti-inflammatory agent. So yung papain that are present in your pineapple is responsible para na, di ba gumagawa tayo ng gelatin, gumagawa tayo ng mga desserts. So, kapag gumawa tayo ng gelatin, kapag fresh ang ating pineapple with the presence of enzyme papain, hindi nabubuo ang ating gelatin because of the presence of papain. Okay, so now, enzyme. So, ang enzyme parang lock and key. Okay? So, this is your enzyme and this is your substrate. Diba? Substrate 1 and substrate 2. Okay? Kailangan mag-fit ang enzyme doon sa ating substrate. Okay? So, parang lock and key. Hindi magkakaroon ng chemical reaction ang any reaction kung hindi mag-fit ang enzyme at saka substrate specificity. Yun ang tinatawag nating substrate specificity. Okay, so ito yon, meron tayong substrate 1 and substrate 2. Parang puzzle. Okay, so dapat yung substrate 1 mag-fit doon sa structure ng enzyme. Okay, yung substrate 2 can also fit doon sa enzyme para magkaroon siya ng product at saka yung enzyme. Okay, so this is the product. And also, yung pagka nagkaroon ng product, okay, so magkakaroon ng enzyme. So hihiwalay yung enzyme doon. Yung enzyme can also be used. Okay, pwede ulit natin siyang gamitin reversibly. Okay, pwede siyang reversible, pwede natin siyang gamitin pa ulit-ulit. Okay, so hanggang sa basta yung enzyme substrate specificity. So dapat specific ang enzyme at saka yung substrate. Okay. So enzyme example. So enzyme natin, example is a pepsin. Okay. So pepsin, Ano ang role ng pepsin? Stomach enzyme used to break protein down into peptides, as I made mention, works at a very acidic at pH of 1.5. Okay, so pepsin yun. So, responsible yung pepsin na enzyme, responsible in breaking down protein into amino acids. Okay, binibreak niya is the peptide linkage between amino acids of the protein. Proteases, this is a digestive enzyme which acts on protein in the digestive system. Basically, proteases also acts on protein, inaatake niya ang protein to break down into simple substances, which is the amino acid. Kung yung pepsin is a stomach enzyme, yung ating proteases can be found in your digestive enzyme. So amylases. So amylases is a family of enzymes which assist in the breakdown of carbohydrates. So amylase natin, like for instance, the salivary amylase can be found in your mouth. So saliva natin yon. Yung saliva contains the amylases. It can break down the carbohydrates into a simple form. Okay, kaya nga yung sinasabi natin, kapag kumakain tayo ng polysaccharide dito sa bibig natin na walang lasa, eventually magkakaroon ng lasa, tatamis, because of the presence of the amylases in your saliva. Okay, breaks down the polysaccharide into monosaccharides, kaya medyo tumatamis. Diba? During the discussion in your carbohydrates, lahat ng monosaccharide are sweetish by nature. So, lipases, a family of enzymes which break down lipids. So, meron tayong enzyme substrate specificity. Kung itong lipases natin is specific only for lipids. Okay? Yung amylases natin is specific for carbohydrates. Proteases and pepsin is specific for protein. Next one. One of the fastest enzyme in the body is the catalase. Catalase break down hydrogen peroxide, a waste product of cell metabolism, into water and oxygen. Accumulation of hydrogen peroxide is toxic, so this enzyme performs an important job in the body. So this is one important enzyme, which is your catalase. That is one that catalyzes or that breaks down, okay, the hydrogen peroxide, that catalyzes or breaks down the hydrogen peroxide, a waste product of cell metabolism. Okay, so all reactants need to have a certain energy before they will react. This is like an energy barrier that it has to overcome before a reaction will occur. It is called the activation energy. So, kailangan meron muna tayong power, the activation energy. Enzymes are organic catalysts kasi nakikita natin ang enzymes natin can be found in the living system. All catalysts lower the energy barrier, allowing the reactants or the substrates to react faster, forming the product. So, without the catalyst, mabagal ang reaction ng any chemical reaction. In this case, nandito tayo sa biochemistry, ang kailangan natin as enzyme is a catalyst, is a biochemical catalyst to speed up chemical reactions. Enzymes do not participate in the reaction. Hindi siya nagpa-participate in the reaction. Nakita mo, in any chemical reaction kanina, di ba? Doon sa chemical reaction natin, humihiwalay yung enzyme at saka yung product. Once na nag-react yung substrate at saka yung enzyme, pag nag-end na siya sa product, humihiwalay na yung enzyme. Yung enzyme kasi will also act again into another substrate. Can be used up into another substrate to form another product. Okay, to form another product. So, this is what we call the enzyme activation. Okay, so makikita natin. So, meron tayong high energy dito, the reactant, and the low energy. Okay, so without an enzyme, the activation energy is high. Okay, the high energy. Okay, so with enzyme, the activation energy required is lower. Okay, so without an enzyme, kailangan natin ng energy required is high. Doon sa enzyme, okay, kailangan the activation energy required is lower. Okay, so nakita nyo dyan, yung high energy, yun, okay? So low energy, so sa reactant, so this is how to explain how enzymes activate yung with regards to the pagkakaroon ng reactant at nagkakaroon tayo ng product with the reaction of enzyme. Okay. Enzymes have a specific region where the substrates bind and where catalysis occurs. This is called the active site. Okay. So, enzymes are substrate specific, although specificity varies from enzyme to enzyme. When a substrate binds to an enzyme active site, an enzyme substrate complex is formed. Okay. So, meron tayong specific site or active site of a particular substrate. So, ibig sabihin, parang doon sa kanina example natin kanina, yung kailangan mag-fit sa active site yung inyong enzyme. Kung hindi mag-fit ang enzyme doon yung sa active site, so hindi siya magkakaroon ng chemical, hindi siya magkakaroon ng reaction. Okay? So, kaya nga meron tayong tinatawag na substrate and specific, okay, or specificity, or enzyme substrate specificity. Okay, so meron lang siyang sinasamahan, specific ang substrate at an enzyme. Okay, so kung hindi siya specific, hindi magre-react yung enzyme doon sa active site mo kasi hindi naman siya mag-fit doon sa active site ng inyong substrate. Parang 'di ba, as I have said, parang lock and key. May particular na key doon sa isang lock. Okay? So, yun yung tinatawag nating Lucknk Model. Okay. So, enzyme active site. So, substrate molecule. Okay, substrate molecule are the chemical that an enzyme acts on. They are drawn into the left of the substrate. Ito yon. So, doon siya sasama para magkaroon siya ng reaction. That is what we call the active site. Okay? So, yung enzyme molecule, okay, this is your enzyme, will be going into that para magkaroon siya ng enzyme specificity reaction. Okay? Kung mag-fit itong enzyme mo doon sa active site, okay, magkakaroon sila ng enzyme substrate specificity para magkaroon ka ng product. Okay. Next one. Okay. So, this is the best explanation of the Lucknk model. The Lucknk model of enzyme action proposed earlier this century proposed that a substrate was simply drawn into closely matching clefts on the enzyme molecule. Ganito lang kasimple. Kung ganito yung inyong enzyme, okay, dapat mag-fit siya dun sa substrate mo. So, nakita nyo dito sa number, okay. So, sa number 2 nyo, sa number 1 nyo is a substrate is drawn into the active site of the enzyme. So, kung fit itong substrate dun sa enzyme, magkakaroon ka ng Lucknk model. Okay. So, dun sa number 2 mo, the substrate shape must be compatible with the enzyme active site in order to fit and be reacted upon. So, magkakaroon siya ng reaction dyan kung magkakaroon siya ng enzyme substrate specificity. And then, yung number 3 mo, the enzyme modifies the substrate. Okay, magkakaroon ka na ng product. Ito na nga yung product mo. Napaghiwalay na natin yung product with your reaction of an enzyme. If this enzyme is your amylases, okay, if this is your substrate is a carbohydrate, okay, magkakaroon ka ng product na monosaccharide or disaccharide kasi na-split up mo yung polysac into its simple form. Okay? Basically, yung enzyme mo hindi magbabago kasi mag-fit ulit yan sa another reaction. Kung enzyme mo is specific for protein, if this is your protein, magre-react sa protein, and then specific, magkakaroon ka ng amino acid. Kung specific ito for lipids, magkakaroon ka dito ng, or if this is your lipases, for example, and this is your lipid, magkakaroon ka ng product ng monomer ng lipids. Okay, the enzyme modifies the substrate. In this instance, the substrate is broken down, releasing two products. Okay, yun nga, yung inaano natin na end product nila. Okay, induced fit model. The most recent study that revealed that the process is much more likely to involve the induced fit. Okay, number one, two substrate, okay, ito yung two substrate molecule are drawn into the cleft of the enzyme. If this is your enzyme, okay, and this is your substrate, the enzyme changes its shape. Kaya nga tinatawag nating induced fit model. Okay, forcing the substrate molecule to combine. Pinagko-combine niya yan, and the resulting end product is released by the enzyme which return to its normal shape, are ready to undergo more reaction. As I have said kanina, hindi magbabago ang inyong enzyme kasi magre-react pa siya sa maraming substrate pa. Okay, in this case, in the induced fit model, the enzyme or the reactants changes their shape slightly. The reactants becomes bound to enzyme by weak chemical bonds. This binding can weaken bonds within the reactants themselves, allowing the reaction to proceed more readily. Okay, so that is what we call the induced fit model. Changing the active sites. Changes to the shape of the active site will result in a loss of function. So pag binago natin ang shape ng active site, magbabago or mawawala ang function ng inyong enzyme. Enzymes are sensitive to various factors such as temperature and the pH. When an enzyme has lost its characteristics, it is said to be denatured. Okay, it is said to be denatured kasi nagbago na siya ng anyo. Some enzymes can regain their shape, while in others, the changes are irreversible. Hindi na mababago ang anyo kasi na-denature na siya. Okay, so let us now identify the effects of temperature on the enzyme action. Okay, so in this graph, meron tayong percent optimum activity into 0 to 100. Meron dito tayong temperature. So, too cold to enzyme to work at 0 degree, ayan, hanggang sa maabot niya yung optimum temperature of the enzyme para magkaroon ng react. Pagkat pataas naman syempre, may buhay yung enzyme, mamamatayan. Okay? Too hot for enzyme to work. Diba? Kaya nga tayo kapag nagkakaroon tayo ng fermentation of sugar, di ba, gumagamit tayo ng yeast. Bakit pinapalamig pa natin yung ating solution, ng ating sugar solution para magkaroon tayo ng fermentation of sugar? Kasi mamamatay yung enzyme na yeast present, the enzyme present into your yeasts, di ba? So, hindi siya mag-work, hindi siya magkakaroon ng fermentation. Okay, mabubulok lang ang inyong fermentation kasi namatay na nga yung enzyme. Okay, so speed up all reactions, but the rate of denaturation of enzyme also increases at higher temperature. High temperatures break the disulfide bonds holding the tertiary structure of the enzyme. Together, they're changing the shape of the enzyme. So this is the activity pagka tumataas yung ating temperature. Okay, so kapag naman masyadong tumaas naman yung temperature, this destroys the active site and therefore makes the enzyme non-functional, as I have said. Okay, so the rate of reaction, okay, so we have the temperature. The curve on the blue represent an enzyme isolated from an organism living in the Arctic cell. Okay, these cold-dwelling organisms are called psychrophiles. Okay, so rate of reaction. The curve in the red represent an enzyme isolated from the digestive tracts of human. Okay, yan yung specific temperature. Okay, so kapag tumaas, di ba? Okay, so at 4 degrees, okay, at 4 degrees, okay, doon sa curves, yun yung isolation of an organism living in the Arctic. Okay, so at 37 degrees, okay, at 37 degrees, doon yun nagkakaroon ng Yun yung pinaka-peak ng isolation ng digestive tract. Okay? So yun yung pinaka-peak nung at 37 degrees, yun yung pinaka-peak wherein digestive enzymes can be reacted into your system. The curved in green represent an enzyme isolated from a thermophile bacteria found growing in geothermal seabeds at 95 degrees. So, yung thermophile, kasi kailangan nila ng mataas na temperatura in order to react. Okay, at 95 degree. Okay, now let's move on to the pH. Okay, what are the effect of pH on enzyme action? So rate of reaction, this is pH 1 to 2, pH 5 to 7. So pH 2 is the peak and pH 7 is the peak. Like all proteins, enzymes are denatured by extremes of pH, acidity or alkalinity. The green curve is for pepsin. Ito yun, pepsin, that digests protein in the stomach. Ito yung pinaka-peak at pH 2. The red curve represents the activity of arginase that breaks down arginine to ornithine and urea in the liver. So, yun yung pinaka-peak at pH 7. Okay, so yun yun na. So, based on acidity and alkalinity of the pH. Next one. The effects of the enzyme concentration on enzyme action. So, rate of reaction, so enzyme concentration. Okay, more enzyme molecules can react with more substrate molecule, so the reaction rate increases. So, nag-i-increase. Once na enzyme concentration increases yung rate of reaction. Okay? So, meron tayong both sides of the graph are increasing because pagka nag-increase yung enzyme concentration, nag-increase yung rate of reaction. Assuming that the amount of substrate is not limiting, an increase in enzyme concentration causes increase in the reaction rate. Okay, so tatandaan na, kapag ka nag-increase ang reaction rate, nag-increase ang enzyme concentration and vice versa. Okay, so again, meron tayong reaction rate, meron tayong substrate concentration, the effects of substrate concentration and enzyme action, assuming that the amount of enzyme is constant, okay, constant assuming that the amount of enzyme is constant, an increase in substrate concentration is causes a diminishing increase in the reaction rate. Okay, nakita nyo, nagkakaroon ng sa reaction rate. So, bumabagal na siya. Okay. A maximum rate is obtained at a certain concentration of substrate when all enzymes are occupied substrate. The rate cannot increase any further. So, kapag ka doon, so, it depends on the substrate concentration. Meron tayong specific or maximum, okay, or maximum reaction rate. Okay, the effects of cofactors on enzyme action. So, cofactors are substances that are essential to the catalytic activity of some enzymes. So, when we say cofactor, para bang, agent siya. Okay? So, parang agent siya, para siyang body-body in order to react. Okay? Cofactors may alter the shape of the enzyme is likely to make the active site functional or to complete the reactive site. Okay. So, siya ba yung nagpapabago ng shape ng enzyme or slightly alter the shape of the enzyme to make it the enzyme fit with the substrate. Okay. Okay. Enzyme cofactor includes coenzyme, organic molecules, or activating ions, for example, sodium ion and potassium ion. Vitamins are often coenzyme. Example is vitamin B1 and vitamin B6. So these are some examples of the cofactors that can be that alter the shape of enzyme para mag-react or mabago or maging functional yung active site ng inyong enzyme. The nature of enzyme inhibitors. Enzyme inhibitors may or may not act reversibly. So, reversible. Okay. The inhibitor is temporarily bound to the enzyme, thereby preventing its function used as a mechanism to control enzyme activity. Reversible yon. Thereby, nagpe-prevent siya ng function. Yung irreversible, the inhibitor may bind permanently to the enzyme causing it to be permanently deactivated. So, ganun yun. Di ba? Parang yun sa COVID natin, kailangan natin ng, di ba, protein yun by nature. So, kailangan natin ng enzyme para magkaroon siya to deactivate it. Para magkaroon siya ng irreversible, okay, inhibitor that may bind permanently to the enzyme causing it to be permanently deactivated. Okay. The nature of enzyme inhibitor. Reversible enzyme works in one or two ways. Competitive inhibitors. The inhibitor competes with the substrate for the active site, thereby blocking it and preventing attachment of the substrate. So kaya nga competitive eh. Nakikikompete itong ating inhibitor doon sa ating substrate, doon sa ating active site to make it para hindi siya to block it and prevent it for the attachment of the substrate. So non-competitive, by the name itself, okay, the inhibitor binds to the enzyme, okay, but not at the active site, and alters its shape. Binabago niyo yung shape, non-competitive. It markedly slowed down the reaction rate by making the enzyme less able to perform its function, or what we call the allosteric inhibition. So, hindi siya actually katulad ng competitive na nakikipagkompetensya that are blocking, okay, from the preventing the attachment of the substrate. Do sa competitive. Sa non-competitive, so inaano lang niya. So, ini-slow down niya yung reaction rate by making the enzyme less able to perform its function. Okay, so yan po ang nature of enzyme inhibitor. Okay? So, for the summary, enzyme works very rapidly and help to speed up biological reactions. Enzymes can be used multiple times. Enzyme can work in both directions of a chemical reaction. Enzymes have optimal temperature and pH that will operate. Beyond these optimum ranges, they will either not work or become denatured or unfolded or break down. Enzymes are usually specific to one particular substrate.